Good afternoon, my name is Raymond Khan and I'll be presenting our enzyme project today. So an enzyme is a biological catalyst that selectively speeds up a biological reaction. It is involved in almost all living organisms. There are three types of enzymes: protease, amylase, and lipase. Protease is responsible for breaking down the proteins into amino acids, whereas amylase is responsible for the breakdown of starch, and lipase enzymes break down lipids. In our own experiment, we're using trypsin, which is a protease. Enzymes are able to speed up chemical reactions by lowering the activation energy required before the chemical reaction can begin. This will allow the reaction to occur faster. And enzymes are important in living organisms because they allow organisms to maintain homeostasis. They are important for metabolic pathways in the body, digestion, and other life processes. Without enzymes, the chemical reactions that occur in the living organism would be too slow to keep it alive. So trypsin is an enzyme that breaks down dietary proteins into individual amino acids and small peptides. This would further allow those amino acids to be absorbed via the bloodstream. So trypsin is active once it is in the small intestine, but is inactive when it is being produced by the pancreas. The question we are answering with our experiment is how salt concentration will affect the activity of an enzyme. We hypothesized that the optimal salt concentration that the enzyme will thrive in is 0.15 molar NaCl. Our hypothesis rationale is that due to the variety of salt concentration in one's diet, we believe that there is an optimal concentration that enzyme will work best in. So for material and method, for our experimental variables, we used 400 microliters of reaction buffer, 500 microliters of substrate, which contains sulfanilamide, and 50 microliters of the enzyme. For our negative control, we replaced the 50 microliters of enzyme and replaced it with a reaction buffer. And we incubated the reactions at 40 degrees Celsius for around 10 minutes. After incubation, we added around 500 microliters of 10% TCA to each sample to stop any enzyme reactions. We then centrifuged these samples at 14,000 RPM for 2 minutes. Once the centrifuge finished, a pellet formed at the bottom of the tube containing undigested proteins, and the supernatant contained the digested protein from the substrate. We then transferred the supernatant from the centrifuge tube to a cuvette so that the rate of absorbance can be measured. And due to the substrate being treated with sulfanilamide, which binds to peptides and makes the digested protein visible in the spectrophotometer at 440 nanometers. For the experiment, we chose the pH and temperature based off what we thought would be the ideal conditions in the intestines. So we chose 40 degrees Celsius and a pH of 8. So we thought if we replicated the environment that the enzyme is found in, it will yield the most activity, leading to better results. We then chose to do 3 trials at each concentration because it gave us more information and removed any outliers that we may see and come across. So we had only one negative control trial and one positive control trial. So we used only one negative control because we found that there was no reason to believe that there would be any change in activity due to the absence of the enzymes. So results. The results we have showed that the average for each concentration stayed within a total of 7% of one another. So as you can see from the trypsin enzyme activity versus the NaCl, we calculated that our t-tests compared to the absorbance levels of 0.15 because the average yielded the highest absorbance. The t-test then showed that there was no significant difference between the different concentrations. There was also a low variability between the trials, which was calculated with the standard deviations, and all trials stayed between 3 to 5%. So as you can see from this is the reaction buffer 0.05, 0.15, 0.3, and 0.6. So we then had the negative control at these 0.20, 0.023, 0.019, and 0.035. And then we had all three of our trials, the averages, as well as our standard deviation in the t-test that we just had explained. So the discussion. The experimental design was not completely adequate to test the hypothesis because there was only one trial for the negative controls. Having only one trial didn't allow us to tell if there was little to no activity. It also didn't give us enough results to compare with the other experimental results for a t-test. The group who also tested salt concentration had somewhat different results than we had. That group had the highest activity at 0.6 molars, while ours was at 0.15 molars. They also had higher differences between their other average results, while ours stayed in between 7% of one another. Overall, the results averages between our groups and theirs was around the same. The differences could have been due to different temperatures that may have been used. The source of variability is very low in the experiment except for the negative control. The standard deviations only made up less than 8% of their averages. Potential sources of variability could be due to one child for the negative control, and other sources could be due to difference in incubation time and pipetting errors. For future attempts, having 3 trials instead of 1 trial for the negative control group could potentially be more beneficial to us. Also, changing the control groups like pH to 9 and maybe a temperature to a higher degree can yield the better results for the experiment. Thank you.